Mr. Keith.
Good morning, my lady. The two witnesses this morning are, please, Professor Stephen Evans and Professor Daniel Prieto Alhambra. If they could be sworn.
Repeat after me. I swear by Almighty God.
I swear by Almighty God
That the evidence I shall give.
that the evidence I shall give
Shall be the truth.
shall be the truth,
The whole truth.
the whole truth,
And nothing but the truth.
and nothing but the truth.
Thank you. Please sit down. Just like to repeat after me. I do solemnly, sincerely, and truly.
I do solemnly, sincerely, and truly
Declare and affirm.
declare and affirm
That the evidence I shall give.
that the evidence I shall give
Shall be the truth.
shall be the truth,
The whole truth.
the whole truth,
And nothing but the truth.
and nothing but the truth.
Good morning.
My lady, if it meets with your approval, I propose to call and deal in detail with Professor Stephen Evans' report first, because he deals primarily with the systems and the processes to do with safety. But where we get to areas on which Professor Prieto Alhambra has opined, I will bring him into the forensic debate, and then we will deal with the majority of Professor Prieto Alhambra's report after that. So perhaps I could commence with asking you both to identify yourselves, and then we'll deal with your qualifications and expertise. For the record, Professor Evans, could you give us, please, your full name?
Stephen James Weston Evans.
And Professor Prieto Alhambra, your full name, please.
Professor Daniel Prieto Alhambra.
Thank you very much, both of you, for the provision of your expert reports. In your case, Professor Evans, dated the 6th of November 2024, and in your case, Professor Prieto Alhambra, September 24. Both are very significant, lengthy documents, and we're very grateful to you for the evident hard work and time that has gone into those reports. May we take it that both reports are the product of your own work, although, as we'll see in a moment, you've been greatly assisted by a number of other professionals and experts, and that the contents are true. I'm sure they are. And dealing with your qualifications, firstly, Professor Evans, your first degree was from Kiel University in physics and chemistry. You have a long list of postgraduate qualifications, but you were, from 1979, a senior lecturer, then the reader, then professor of medical statistics at LHMC, the London School of Hygiene and Tropical Medicine. You were a member of the Working Party of the Royal Statistical Society on Statistics and Drug Regulation. You then left the London School of Hygiene and Tropical Medicine to become head of the epidemiology unit at the UK Medicines Control Agency, now the MHRA. You were an honorary professor of medical statistics at the London School of Hygiene and Tropical Medicine, as well as LHMC. I'm sorry, I think I've referred to LHMC as the London School of Hygiene and Tropical Medicine, but it is in fact the London Hospital and Medical College. You became Professor of Pharmacoepidemiology at the London School of Hygiene. You were on you were on a number of EU committees on medicine safety between 2006 and 2018, a member of the WHO Global Advisory Committee on Vaccine Safety, chair of the Royal Statistical Society Medical Section, and a host of other qualifications. Importantly, for the purposes of the subject matter of your report, did you also have considerable involvement in the Open Safely data program, the collaboration from 2020, and also during the course of the pandemic, did you give some limited advice to the Vaccine Effectiveness Expert Panel in the Cabinet Office and the Vaccine Effectiveness Expert Working Group organised by Public Health England?
I did.
I think you participated as a volunteer in one of the trials, is that right?
I did, in the AstraZeneca trial.
And are you also, as it happens, the convener of the statistics expert group at the Infected Blood Inquiry under Sir Brian Langstaff? Yeah. Professor Prieto Alhambra, you, is this right, are an expert in pharmacoepidemiology. What is the discipline of pharmacoepidemiology succinctly?
Yeah, it's a specialty of epidemiology where we study the use, the safety, and the benefits of medicines and vaccines in the general population or in wide populations.
And is the element of epidemiology in pharmacoepidemiology reflective of the fact that you're dealing with the impact of medicines and vaccines and their safety at population level? You're looking at trends, you're looking at the reality of the impact of the medicines and the vaccines.
Indeed, that is right, and also the use of observational data, typically rather than randomized controlled trials.
Have you trained and practiced for many years as a medical doctor, both in general practice, also in musculoskeletal sciences and rheumatology? And therefore, you've got considerable experience of the clinical diagnosis and management of some of the conditions upon which you opine in your report.
That is correct.
You very properly raised with the inquiry the fact that during the pandemic, you spent some time supervising a PhD student who was embedded within the UK Health Security Agency. You also did some consultancy work providing AstraZeneca with general advice on how to design observational studies for the monitoring of vaccine safety. But you're at pains to point out, quite rightly, that you didn't conduct any studies for them or studies funded by them in relation to COVID-19 vaccines. And you obviously, therefore, weren't involved in any shape or form in AstraZeneca's take on the production, manufacture, authorization of their Oxford AstraZeneca vaccine, or the issue of TTS or thrombocytopenia, and so on.
That is all correct.
In your case, Professor Prieto Alhambra, have you been greatly assisted by a number of co-authors, all of whom I think are at the Nuffield Department of Orthopaedics, Rheumatology and Musculoskeletal Sciences at Oxford? Dr. Anita Jodike, Dr. Edward Byrne, Dr. Z and Dr. Lee. They've all helped you in relation to some parts of your report and provided information and data, but the report is your work.
Indeed, and with the review of the literature, which was substantial.
With the review of?
The literature, the published evidence.
Ah, yes, well, we'll come to that in a moment. Yes, there we are. So we're going to start, please, Professor Evans, with your report, because you focus on the systems and the processes, and we need to address those first and get them in our minds. And we're going to start with the issue of trials. The scientific process of trialing vaccines starts in the laboratory, does it not, with what are known as in vitro studies. What are they? Are they clinical trials carried out on people, or are they laboratory tests?
They're laboratory tests.
And can a manufacturer hoping to produce a medicine, whether it's a vaccine or not, proceed to the clinical trials without having passed successfully through the in vitro, the laboratory aspect of the trial process?
They have to go through that and through animal studies as well.
And do many products, many studies end at that stage and not proceed through to clinical trials?
Very large numbers.
And in very rough terms, what is the purpose of the preclinical trial process? Does it focus on safety? Does it focus on whether or not the product works, whether or not it has unexpected side effects? What is the purpose of the laboratory angle?
The laboratory angle is really to understand the way it is likely to work in the human body, and in particular, what kind of dose of the product is likely to, what sort of dose range among the vast possibilities. It doesn't really look at efficacy or safety at that stage.
And therefore, only if the tests are satisfactorily completed at that stage may the product then be tested on humans.
Yes, they will look in animals for things like cancers that might only take a few weeks to develop in an animal, whereas in a human being it might take tens of years.
And then the clinical study phase, does that comprise 3 phases, in fact, pre-authorization, phases 1, 2, and 3?
Yes, those are labels that there are overlaps between them.
Is phase 4 a term given to such trials as may be conducted post-authorization?
Yes.
Focusing, of course, on clinical use, because post-authorization,
yes,
the medicine will be in general use or in use, and therefore can be given to patients.
Yes.
All right. So just focusing then on phases 1, 2, and 3, could you broadly delineate between them, please, Professor? Phase 1, what does it focus on?
Phase 1 is going to focus on whether we've got the dose right, usually in healthy volunteers. Particularly for drugs, you will not be using people who've got the disease that you're targeting, but for vaccines, healthy people are your target. So you test whether the dose seems to cause a really nasty adverse effect or not, because you won't necessarily know that beforehand.
Phase 2?
Phase 2, you're moving up the scale and you're beginning to be able to understand whether the drug has the effect or the vaccine has the effect you expect it to have. And you will generally be measuring biochemical things within the body's response, the so-called immune response to the vaccine, to see whether that is happening in the way you expect it to in phase 2. And again, ensuring that you narrow down the dose that you will have started with a potentially very wide dose range, and you gradually narrow it down to the optimal dose range.
So phase 2 proceeds on the premise that the vaccine has broadly the desired effect, but it's a question of measuring more precisely its impact, particularly for the purposes of deciding dose.
You're measuring the effect on biochemical things happening, the immune response in the body, rather than the response to the virus.
At the phase 2 stage, what size in general terms might the participant group be?
Typically in the tens to perhaps in the hundreds.
And in all the phases, is the closest attention paid, of course, to the consequences of providing the vaccine or medicine, and I emphasize vaccine is just a form of medicine, to the participant. So
yes,
these are very, very closely scrutinized procedures.
Yes.
Phase 3, what is the difference with that? Well, what does it focus on?
For vaccine trials, you then will be interested in whether people get the infection that you're trying to prevent. So if it's COVID-19, you actually test them to see whether they get that, rather than simply whether their body is producing antibodies.
So whether it works in practice?
Whether it works in the way that will make a difference to the disease in the population.
And is safety and the possibility of side effects something that is even more closely scrutinized in phase 3?
Yes, I would say they're probably of equal scrutiny.
And in terms of participant numbers, what sizes may a phase 3 trial amount to?
In drugs, it will still typically be in the hundreds or perhaps into the thousands. But with vaccines, particularly more recent vaccines, it's in the thousands and 10,000s.
You refer repeatedly throughout your report to what is elsewhere known as the gold standard of trial procedures, namely a randomized controlled trial. Is that randomization a reflection of the giving on a random basis of the proposed medicine to some participants, but a placebo or a control to others?
Yes, the idea is that you can be certain within probability limits that the two groups or more than two groups are the same in every respect, whether you can measure those things or not.
And so the outcome, the figures, the results, will be indicative of the impact, they will reflect the impact of the proposed medicine.
Yes.
Because that is the only basis upon which there can be a difference of outcome.
Exactly.
And how important is it that a randomized controlled trial approach is applied in terms of eliminating bias or rogue figures or erroneous outcomes?
It is absolutely vital, because otherwise you don't know whether the people who get vaccinated are the same either in their characteristics or in their behaviour as those who are unvaccinated. So when I was a participant in the trial, I didn't know what I was getting, and I thought I was getting the placebo because I didn't have any strong reactions. In fact, I got the real thing. But had I known what it was, I might have changed my behaviour.
And you might have misreported perhaps even your symptoms or any kind of reaction.
I might have behaved in different ways.
That was a blind trial because you didn't know whether you were receiving the vaccine or a placebo. What is a double-blind trial?
A double-blind trial is where the participant and the investigator giving it to them do not know.
So who knows whether each participant has
received? It will be a statistician like me who's hidden away the list of which treatments it is, and the pharmacist or the manufacturer who's got the code that says bottle number 23 has the real thing.
I now simply can't recall whether I've asked you to opine on the general size of phase 3 trials.
Yes.
I have.
Different for drugs and different for vaccines.
Thank you very much. In the context of the 3 COVID-19 UK vaccines that we're focusing on, and they are the AstraZeneca, Oxford AstraZeneca, the Pfizer-BioNTech, and the Moderna vaccines only, were there phase 3 trials in majority or exclusively randomized controlled trials?
Yes.
Were they double-blind or blind?
They were in some instances double-blind, in some instances single-blind. That was that the investigator did know in principle what the whether it was the real vaccine or a control being given.
With that size of phase 3 trial in mind, why can they not be even larger?
The problem is that you will run the trial, and generally there will be a data monitoring board that will look at the key outcomes. In the phase 3 trial, it will be clinical infection. And you will ethically need to stop the trial or stop recruitment to the trial when you have convincing evidence of efficacy, because otherwise you are subjecting your control group to no treatment, or a treatment that is we know would be ineffective, when we know that there is an effective alternative. And that is regarded by most people as unethical.
Essentially, it's unfair on the participant to oblige them to continue to take part in a trial that's ongoing when it's obvious that the vaccine which is being trialled can actually provide a real-world solution to the terrible pathogenic disease.
That's a subtly different point about the continuation. It's a question of whether you recruit new people. Is my new person going to come in and be likely to have the placebo when there is an effective treatment? And that would be regarded as unethical generally.
We're going to turn a little later in more detail to the very important issue of whether it is at all likely that very rare, serious side effects will be revealed by a trial consisting of participants to be measured in the tens of thousands. But I will come back to that. In your report, you set out much about the way in which major trials are conducted, and you refer to the existence of something called a Data and Safety Monitoring Board. You've already referred to it already this morning. What is that?
It's a group of independent people who will have access to the data that nobody else has access to, and will look at the results and say, for example, is there an adverse effect occurring with the vaccine that is occurring at such a rate that it suggests a significant harm? And if that were a serious adverse effect, then they may want to entirely stop the trial. They may pause the trial if there is just slight evidence that there could be a problem to investigate things more carefully. And the Data Monitoring Board will be an advisor to the investigators who don't know what the results are showing, saying, is it efficacious or not? Is it ethical to continue randomizing new patients to this trial?
And what sort of people generally make up the Data and Safety Monitoring Board? You say it's independent, but...
They're independent, but they're typically about 5 people, maybe 6 people. They will generally have a very experienced clinician who is chair of it, but occasionally it can be a statistician who is chair of it. I've chaired data monitoring committees in the past, but you require somebody who, as Professor Sir Chris Whitty said, it's important to get the people right, and getting the people right is a key component. So you have people with experience in vaccines, but you will also usually have a statistician who can help you with the numbers.
You also refer to a different body, a trial steering committee, which appears to conduct the trial or be responsible for the conduct of the trial. By contrast, is it staffed by not just independent scientists, but investigators and representatives of the manufacturer?
Yes.
And is it that body, the trial steering committee, which, as it says on the tin, steers the progress of the trial and hopefully to its conclusion? As a general rule, indeed as a principle, will there ever be a representative from the manufacturer, the investigator, or somebody else in the company present at the Data and Safety Monitoring Board when that board is discussing matters such as unblinded data, i.e. who gets the placebo or the vaccine, and talking about highly sensitive issues concerning them?
I cannot say that never happens, but I'd never participate in a board in which the manufacturer was present.
So
it must be extremely rare if it does occur. I can't say never.
And the presence and the role of the Data and Safety Monitoring Board is reflective, is it not, of the extraordinary importance which is placed on safety and monitoring safety and observing the possibility of side effects in the course of the trial process.
Yes.
Right. Elsewhere in your report, page 31, in fact, but we needn't go to it, you refer to the fact that randomized trials for the COVID vaccines were conducted in many countries and many regulatory authorities were involved. The 3 COVID-19 UK vaccines were trialled in some cases in the United Kingdom and abroad, and I think in one of the cases exclusively abroad. But between the trials conducted in the United Kingdom and those conducted abroad, were there any significant differences in terms of the structures that were applied, the presence of a data and safety monitoring board, the use of blind and double-blind randomized controlled trials, and the safety setup for the regulation of those trials?
No, they're all done in exactly the same way according to a very rigidly set out protocol. It is the different centres that exist within a single trial, effectively.
And we'll hear a bit more from Professor Priyata Alhambra a bit later, but were in fact some of the thresholds, some of the scientific requirements, for example, the degree of confidence in the efficacy of a vaccine, the way in which the products were to be trialled and investigated, agreed between certain Western European, American and Canadian regulators?
Yes, international regulators agree.
International regulators. So the whole process of the safety monitoring, the trialling and the authorization of all these vaccines was very much an international enterprise.
Very much.
Regulated nationally, but based upon agreed structures, approaches, standards and thresholds internationally.
Yes.
All right. Elsewhere in your report on the subject of trials, you refer to the trial sizes, and you make the point, and you've already made it this morning, that once efficacy has been shown, continuing to recruit sufficient numbers would be unethical. What determines the precise size of the trial in respect of a particular medicinal product or vaccine? We note that although all trials were I think, between 20 and 30,000 at phase 3 level, some of them had more trials in more countries than others. Well, what determines that?
There are practical considerations as to how many countries are involved, but what you want to ensure in terms of vaccine trials is, in phase 3 in particular, you're trying to prevent the disease. So if the disease is not occurring, if the virus is not circulating in a country, then you don't want to do a trial in that country. You want to do the trial in the place where the virus is circulating at a high level, so that you get enough people who, on the control group, sadly, will get the infection. And you then hope that the people on the vaccine will not get it. And you need to have sufficient numbers that say, I've got convincing evidence that there is a real difference here and that the vaccine is effective.
And in your professional opinion, were the numbers involved in each of these 3 vaccines at the requisite level?
Absolutely.
Efficacy, the effectiveness of a vaccine. Plainly, there is little point in developing and manufacturing a vaccine that is safe but not effective.
Absolutely.
It serves no purpose. Which part of the clinical trial process is efficacy generally determined?
For example, in the trial I was involved in, then every week I had to take a swab from my throat and my nose and send it in the post to a laboratory to see whether I had had COVID infection. In other cases, some of the trials would only do that for people who had felt ill and who might have COVID. And so you had to record whether somebody got the infection or not, or whether, in the other trials, it was a clinically relevant infection.
Presumably, a vaccine, as with many medicines, will have an impact on the human body at a number of different levels. In the context of a vaccine, what a vaccine in general terms tries to do is to provoke the body into producing antibodies to fight the infection. But the body's antibody system is no doubt a very complex one. I think there are B cells, T cells, and any number of different levels of response. Is the nature of that response clinically examined in great detail as part of the clinical trial process?
Yes, and it's important, as you say, that the vaccine is there to train the body to provide a means of fighting the virus. A drug doesn't do that. A drug attacks a virus or some other element of the disease directly. A vaccine does not have an effect directly on the virus.
It can't be very straightforward to measure the antibody response of the human body.
It's not, these days, it's not that difficult.
It's not that difficult. But is there nevertheless a system by which what one can say how confident one is as to the degree of effectiveness or efficacy?
When you first start with a new vaccine, you aren't absolutely sure that the antibodies you measure will reflect the clinical reality of preventing clinical disease. But after you've done a few trials, you then find that there are measurements of antibodies that will predict the clinical disease. And those are the ones that you concentrate on measuring in the future.
And coming back to the issue of what was agreed at the international level, was agreement generally reached as to the process by which degrees of confidence could be expressed or had to be expressed publicly about the degree of effectiveness?
Yes,
so we can see in the paperwork vaccine X is 80% effective with a confidence level of between X and Y.
Yes, and the idea is that when you didn't know anything, you kind of assumed that it would be similar to a flu vaccine effectiveness. And so most of the regulators required at least 50 and possibly even 60% vaccine efficacy. But you needed to be sure that it wasn't below a certain level. And in Europe and America, they had slightly different views on how low the uncertainty could be.
Safety. Conceptually, is safety something that is open to objective measurement?
It's very difficult. Safety is a euphemism, I think, for harm. We can only measure harms, and safety is the relative absence of harm. So we look for harms. And when we start out, we think there will be a certain kind of harm for a vaccine. Most people will get a sore arm when they get the injection. Some will get a headache. Some will get a slight fever. But you will check very carefully that there aren't very many people getting a very high fever, 39 degrees or 40 degrees, and potentially causing serious illness. And so you check very carefully for those things.
So it simply cannot be said in respect of any product, let alone a vaccine, it is safe or it is not safe. What is required to be done is to analyse the relative safety of the product by examining the benefit and comparing it to the risk.
Absolutely, and this also develops over time. Our knowledge basically increases. I've talked about this as a map, and that we're trying to explore the entire territory of the effect of a drug. And at first, we only know a little bit about that map, but gradually, as we use it more, have bigger trials, and then start to use it, we will extend our knowledge and will have mapped the knowledge of that area of the drug.
And is that why there are what is known as pre-authorization safety studies and post-authorization safety studies? Because after authority is given and once the product is being rolled out, there is a flood of new and important data concerning the clinical use of that product in real-world conditions.
Yes.
All right. And is that why what's known as the post-authorization monitoring process or safety process is so very important?
Yes.
I think we've seen somewhere that within 2 days of the rollout of the Pfizer-BioNTech vaccine in the United Kingdom, there were more vaccines delivered than there had been in the entirety of all the preceding clinical trials.
Yes, I believe that to be true.
Right. All medicines, including vaccines, may give rise to a side effect. That is to say, something that's undesirable, not what is intended.
I know of no effective medicine that does not have unwanted effects, usually adverse.
We've seen in the paperwork references to a number of descriptions and number of terminological phrases: adverse events of special interest, serious adverse reactions, adverse effects, adverse reactions, adverse drug reactions, and suspected unexpected serious adverse effects. You won't, I think, agree with this proposition, but by and large, we're talking about side effects.
Yes.
Right. Coming back to the trial process, if a side effect, that is to say something that is not sought, it's not what the purpose of the product is, it's undesirable, if it is rare or very rare or extremely rare, and the MHRA defines each of those words by reference to the statistical likelihood of them occurring, 1 in 10,000 for rare, I think 1 in 100,000 for very rare, and over or less than, fewer than 1 in 100,000 is generally regarded as being extremely rare. If a side effect is rare, very rare, or extremely rare, is it likely to appear in a trial consisting of around 30, 40, 50,000 participants?
Simply put, the very rare and extremely rare will not be seen even in the trials generally, unless by chance.
Because obviously, if a side effect will only appear 1 in every 100,000 cases, a trial of 50,000 will not reach that point at which statistically the occurrence is likely to occur.
Yes, we can say what the probability of having 1 in that 50,000, even if it occurs generally at 1 in 100,000, in the 50,000 it might occur, but you wouldn't get, it'd be very unlikely that you got 2 occurring in 50,000.
Moreover, and perhaps even more, it's even more difficult a concept, if a person takes a vaccine, receives a vaccine, and there is then a medical condition that becomes apparent, a reddening of the arm, or some medical condition, the issue immediately arises as to whether or not that appearance is coincidental or was caused by the taking of the vaccine, or, I suppose, might have been the result of COVID-19 itself in this case, because a person might be infected with COVID unknowingly and then be vaccinated. So can you ever immediately tell from the mere existence or presence of a medical condition that it was necessarily caused by the vaccine?
Very difficult indeed in individual cases, and that's why we have a randomized trial in which we can have a large number of people, and when there is an excess in the vaccinated group, when we do the statistical analysis on it, we can find that there is convincing evidence. And the randomized trial means that the other factors that might explain it will not be a possible explanation. Whereas in observational studies, it is much more difficult.
Presumably, there are some responses to the taking of a vaccine in particular, which has to be injected, known as reactogenicity or conditions associated with reactogenicity, which are obviously connected to the vaccine because they might be a reddening of the arm or having, I don't know, flu-like symptoms within an hour of taking the vaccine. In relation to that sort of side effect, are they generally ever severe or serious?
Almost all vaccines, if they've got as far as being trialled in phase 3, will not have a high rate of the higher, more severe reactions. But that is what the phase 3 trial will be looking for, to check that there isn't a high rate of the more severe forms.
Can you say whether or not the vast majority of side effects or reactions to the vaccines in the phase 3 trials were of that type? Mild,
Yes.
Momentary, reactogenicity type reactions?
Yes.
And how long, in general terms, do those sorts of reactions or conditions take to appear and disappear?
They take generally from hours to a very few days, generally less than a week.
What can be done about a condition or a reaction that may in the human body take weeks or months or maybe even years to develop and therefore appear?
For weeks and months, the trials will go on following up people. And even when a trial stops recruiting, and as in the case of the AstraZeneca trial I was in, the licensed vaccine became available, and I asked to be unblinded to see whether I had. But I was continued to be followed up for another year or more to see whether there were adverse effects of any serious nature.
And what can further or moreover be done about what is known as the multi-hit impact? That is to say, a person taking a vaccine and suffering already from other conditions, comorbidities perhaps, or being subject to environmental factors, which means that the combination of taking a vaccine alongside their pre-existing or to be developed condition externally brings about an unexpected or unlikely consequence.
These are the very rare reactions that we will not be able to study easily in a randomized trial.
And so what is done about them in terms of post-authorization monitoring?
Well, we need to have very large numbers of people vaccinated, and we need to see what their health effects are, both immediately after and also months and perhaps years after the vaccine.
And in light of all that, do you conclude, for the record at page 16 of your report, that whilst it is relatively easy to detect frequent and rapid onset adverse effects, it is much more difficult to detect rare and serious or delayed side effects?
Yes.
And that is why so much scrutiny must be paid to the post-authorization, post-marketing surveillance, as it's known. Quality we needn't, I think, spend a great deal of time on quality. It's not within the scope, in fact, of this module. But presumably, a significant part of the authorization and the trial and the checking processes, as well as the regulatory scrutiny, focuses on quality. So not just effectiveness or safety, but whether or not it's good quality.
Yes.
And on that point, does each regulator, and including the MHRA, test medicinal products, including vaccines, for their quality?
Yes.
And is there a process by which the laboratories, in the MHRA's case, what were known as the National Institute for Biological Standards and Control laboratories, test each batch?
Yes, they're the world leaders in the field.
Can that testing test for safety and effectiveness, or can it only test for quality?
It can only test for quality, but some aspects of the quality will lead to adverse effects. Impurities may well lead to infections.
And just give us some idea of the scale of this, please, Professor. How big is a batch? How many doses does a batch have?
Well, with vaccines, a batch is made as a biological process rather than a chemical process. And so they're rather variable in size. I'm not an expert in that field, but I would understand that typical batches will have between a few thousand doses and a few 100,000 doses. It doesn't have millions of doses generally, nor does it have tens or hundreds, but it's thousands to tens of thousands. And probably, typically, I would have thought about 50,000 doses, but it may be rather larger. I also understand, but as I say, I'm not an expert in that area, that as the experience with the manufacturing process improves, then the batch size may become steadily larger.
Do you understand, and there's clear evidence that the MHRA's laboratories test every single batch, that the batches that are tested are the product of the same manufacturing process in respect of each vaccine? So if you test every batch, you haven't got a position in which half the batches are being made by an offshore different manufacturing process than the other half.
They're meant to be made under exactly the same process, but it's a biological process, and that's why, particularly with vaccines, it's important to check their quality.
And presumably, the batch testing is something which is also open to international regulation and agreement and scrutiny, and therefore the MHRA's processes for batch testing would be very much aligned with the near identical or very similar processes being conducted by other regulators.
Yes.
Does it follow that the UK COVID-19 vaccines would therefore have been tested, because they were rolled out across the world, by any number of different regulators?
They may well have been, yes.
The authorization process, please. All countries, by and large, well, I've just departed from my own question. Do most countries regulate the marketing and supply of medicines?
Yes.
In the case of the United Kingdom, and it's the whole of the United Kingdom, is it the MHRA?
Yes.
The MHRA is nominally part of government, but is it in fact an independent regulator?
I think they would fiercely
They would object to that description.
object to the idea that they were part of government. They are.
Well, they're part of the state, maybe.
They're part of the state, but they are very much independent.
They, I think, actually, normally they regard themselves as being part of the DHSE, but yes, they.
Yes, administratively they come under the Department of Health, but this is a separation of powers.
So they're operationally.
Their operation and their powers are independent of government.
Yes. And in the case of the UK COVID-19 vaccines, are you aware of how they ramped up the number of staff and the scope of their functions and what they were doing in order to be able to deal with the unprecedented authorization process.
Yes.
You've already mentioned along the way something about the skill and the standard of the MHRA. Where does it stand in world rankings in terms of skill, diligence, robust overview?
I think in terms of its size, it is certainly in the top 5. At one stage, I think it might have been regarded as the world's premier regulator, but probably most people would regard the FDA in America, the Food and Drug Administration, which is a massive body with enormous amounts of funding, as being the world premier regulator.
Does, in fact, the MHRA not just regulate and authorize medicinal products and medicines in the United Kingdom, but scrutinize and check and study medicinal and products and medicines from elsewhere in the world, and I think provide semi-regulatory or scrutinizing processes for other people?
Yes, though the European Medicines Agency does rather more of that.
All right. And we'll hear more from Dame June Rain later, but the MHRA is greatly assisted, is it not, by what is known as a committee known as the Commission on Human Medicines, which provides expert advice, and we have seen from the paperwork that in relation to every aspect of safety regulation in this case, there are very, very learned, detailed papers and produced and meetings held in respect of giving advice to the MHRA on safety issues. How important are the expert working groups that fall within the scope of the Commission on Human Medicines?
Inevitably, any finite group of people can't have all the expertise, and so having expertise for particular areas, such as vaccines, there won't, on the main CHM, it won't have all vaccine experts on it, but the vaccine working group is a specialist group, and they will all have expertise in vaccines. So it's a way of broadening the depth as well as the range of expertise available to the MHRA.
When the MHRA looks at safety and quality and effectiveness for the purposes of deciding whether to authorize a medicinal product, is it concerned with cost or the practicalities of delivery?
No.
In this case, in relation to the 3 COVID-19 UK vaccines, the MHRA authorized each vaccine with varying conditions.
Yes.
So the MHRA cannot just authorize a vaccine, it can put conditions on
Yes.
the scope of the authorization.
Yes.
And by and large, we need to look at them in detail. There were conditions placed by the MHRA in relation to age, in relation to dosage interval, and in relation to whether or not certain sectorial groups should be, subject to the advice of the JCVI and the government, offered a vaccine.
Those would be called the indication, and they do that all the time for all medicines. There may be additional conditions when the knowledge on something is rather more limited, and they specify that extra studies must be done, and it requires those to be done before they get a full marketing authorization, and that specific post-authorization safety studies may need to be carried out.
And there's a significant section in your report which emphasises that there are bodies across the United Kingdom, NICE in England and Wales, the Scottish Intercollegiate Guidelines Network in Scotland and the Scottish Medicines Consortium, and in Northern Ireland, NICE is applied through their devolved structures, which are concerned with the issue of whether or not a particular medicinal product should be offered to members of the public clinically. But they have no role whatsoever in the issue of safety, do they?
No.
The authorization in relation to the UK vaccines was granted under what's known as Regulation 174 of the Human Medicines Regulations. A number of people have observed that that is a UK legal provision. It's a UK Regulation 174. And it is not the legal structure which was and is applied at the European level, at the European Union, of which we were part until 11 p.m. on the 31st of January 2020. Is the regulatory system under 174 in fact derived from a European directive or regulation?
Yes, it was a European regulation, effectively, that was allowed under European law in public health emergencies for any member state to take their own action for a public health emergency. And Hungary did the same, and they were part of the EU and still are, for the moment at least.
And Regulation 174 existed prior to our departure at the end of the transition period?
Yes, it was under the 2012 law.
And it continued to exist following our departure?
It did.
Northern Ireland, as it happens, remained after the transition period part of the European medicinal regulatory structure. But did Regulation 174 nevertheless apply to Northern Ireland and was available to be utilised in the context of Northern Ireland?
That is my understanding. I'm not a lawyer. But I think one of the other things is that there is a European procedure, and the Pfizer vaccine was authorised under European law prior to the end of 2020. And the consequence was that the UK had a marketing authorisation under European law that was then carried over into 2021. So it's a very complex legal issue caused by Brexit in that instance.
But because of the use of Regulation 174, a consistent approach could be applied in terms of the level of scrutiny and legal authorisation across the whole of the United Kingdom. And secondly, was there any difference in substance?
No.
Between the UK regulatory authorisation process and the Europeans?
No.
Finally then, on this topic, when authority came to be considered in respect of each of these vaccines, it's plain from the evidence that an issue had arisen in relation to AstraZeneca in July and September of 2020 of a very, very small number of cases of what is known as transverse myelitis appearing. The existence of those handful of cases paused on both occasions, the trial process, but they were recommenced, and they didn't prevent the authority being given ultimately for the use of the vaccine because, and we needn't go there, it's clear that the MHRA and the CHM, the Commission on Human Medicines, determined that they were not causatively the result of the vaccine. Putting that issue aside, did any vaccine manufacturer in any of these three cases report during the trial process a SUSAR, that is to say, the adverse effect, the suspected, unexpected serious adverse effect?
Adverse reaction, yes.
Adverse reaction.
As far as I know, they did not.
Right. And is it your opinion, therefore, page 32 of your report, that the authorisation process in the United Kingdom was appropriate to the circumstances, was based on a great deal of data, and in fact was completely aligned to the approach to authorization considered and granted in other countries.
Yes, I would agree. And even if there had been a few SUSARs, that wouldn't have affected it.
Do you know of anything that the United Kingdom missed in comparison with other countries at that time?
No.
It is well known that in order to apply a more expedited process, the MHRA altered its procedures for the receipt of data in relation to the trial process and in relation to the application for authorization.
Yes.
So in relation to the trial process, it allowed data to be provided on a rolling basis rather than delaying the whole system by waiting for the manufacturer to produce all the data at the end in one fell swoop.
Yes, it's usually in the manufacturer's interest because they have to pay a big fee when they ask for an authorization. And so they won't want to submit until they're sure they've got a product that works, because it's going to cost them a lot of money to have it assessed.
In relation to the authorization process, was there similarly a rolling review whereby, as a result of very close daily, hourly liaison between the MHRA and the manufacturers, the data was constantly provided rather than being delivered in one fell swoop?
Yes.
In your opinion, did that more expedited and tweaked process have any impact at all on the degree of scrutiny, on the MHRA's understanding of the levels of safety in the clinical trial process, or in relation to the authorization process?
No, I think the scrutiny is likely to have been greater than in a single lot.
Could you elaborate on that? We, of course, were dealing with the trial and authorization process for vaccines, which were subject to incredible public scrutiny. I mean, it was a matter of the great importance to everybody. I mean, COVID presented almost, I suppose, an existential threat. Can you express a view as to the reality of the degree of scrutiny and care and attention that was applied by regulators across the Western world?
I think there's no doubt that they were aware that if they authorized a vaccine that was bad, either in not being effective or having serious adverse effects at a sufficient rate to make the vaccine regarded as useless, they were in real trouble. And I think they are public spirited, and they worked every hour there was in order to carry out the assessment. And by doing it in this rolling way, they became very familiar with the data. They were doing this almost exclusively. There was almost nothing. They weren't doing work for the European Medicines Agency at the time, and so you'd have to ask Dame June Rain, but I think that virtually everybody in that agency was concentrating on doing it, and so the scrutiny was very intense indeed.
Professor Prieto Alhambra, we're at the point where your report, which deals largely with the more forensic issue of particular side effects caused by the vaccines or associated with the vaccines, or which coincidentally followed the receipt of the vaccines. But nevertheless, you've expressed some views on in an overall sense on the regulatory and the authorization process. Paragraph 3.16 of your report, you say, it is my view that the modifications made to the safety regulatory system in response to the pandemic did not negatively affect the ability to effectively identify and respond to safety issues. By that reference to modifications, are you referring to the way in which data was received in respect of the trials, and also the way in which data was received in relation to the applications for authorization?
Indeed, yeah.
You too make the point that, of course, given the scale of the trials none of the potential serious adverse effects which were subsequently identified as being associated, that is to say, without expressing a view on whether they were causatively connected to the vaccines, none of those were known about at the time because they were all too rare to be detected even in a very large clinical trial.
I think Professor Evans explained that elegantly with numbers.
So you agree with what he said in relation to that?
Professor, you have a very soft voice. I don't know if you always have a soft voice when you're talking to your students, but you do here. If you could speak up, I'd be really grateful.
I will do. I am.
Obviously, that observation must exclude, of course, the bringing to the attention of the MHRA and the CHM in July and September 2020 the handful of cases of transverse myelitis in relation to AstraZeneca. But that was a condition at that time which was presumably addressed in very great detail. Was a view ever reached as to whether or not a causative connection could be established between that handful of cases of transverse myelitis and the vaccine? Or was it coincidental or perhaps associated with COVID-19 itself?
I think transverse myelitis is one of these AESI, adverse events of special interest, that we know from previous experience with other vaccines could be potentially related to vaccination. So it was greatly monitored very closely by the MHRA and any other international regulators. Despite that, the quality of the evidence on a causal association is low, maybe moderate. There's a number of studies, some of them good quality, with inconsistent results.
So there is, and we'll look in more detail at the nature of the literature review that you did, but without going into in detail, your overall view was that there is material in the public domain by way of scientific literature which provides low quality or low to moderate quality evidence for the proposition that there is an association between vaccines and transverse myelitis.
Indeed, that's our conclusion.
Right. I'm afraid that's all I'm going to ask you for the moment, Professor. Right, back to Professor Evans. Could we now spend a little time, not very long, dealing with the general obligation on manufacturers to tell regulators any information in their possession which relates to the safety of the product or which might impact upon post-authorization, the conditions of the authorization or the grant of authorization itself. What can you say about the general extent of the obligation on manufacturers?
It's a legal requirement.
Does that legal requirement extend to, well, how far does that legal requirement extend in terms of information relating to the safety of the product under a manufacturer? Is it anything to do with safety, or is it anything which might impact upon the grant of authorization? How is it assessed?
I think that one has to realise that it isn't the manufacturer who is really doing the trials. It is being done in hospitals or in general practices around the world.
Well, that's phase 4.
No, but even in phase 3, I was recruited through Southampton University Hospital, and so I was studied within an academic centre. And so even in phase 3 trials, they're being done with real patients in the real world, but they're under very controlled conditions. And so those clinicians know that they have an absolute requirement to report adverse effects to the investigators, who then have to report it, and it's reported on to the data monitoring board and to the manufacturer. And so the whole process means that if Stephen Evans got a really severe adverse reaction, there will be a nurse who starts with it and then a doctor and so on, and it'll eventually end up at the MHRA through at least one mechanism, and very often there's multiple mechanisms whereby it can reach the MHRA. So there is very considerable pressure on companies to keep to any legal requirement they have. It's possible that they will interpret the law in a slightly different way in different circumstances. But generally, my experience is that they're very good in that.
And so just that we're clear about this, and I went awry my question to you. The phase 4 trials focus on the receipt of a vaccine or a product in clinical use. So
yes, once it's been authorised, that's the point.
It's been given to the public.
Yes.
But hospitals and academic institutions and so on and so forth will encourage members of the public to participate in the phase 3 trials, pre-authorization,
exactly.
Which is why members of the public
yes.
would receive the vaccine.
Exactly.
Right. And I won't ask you the detail of it. We'll deal with it with Dame June Rain. But a manufacturer is obliged to produce a number of documents throughout the trial pre-authorization stage by way of case safety reports, update safety reports, periodic safety update reports, and make provision for something known as risk management plans. That's to say how they intend to go about assessing and monitoring safety post-authorization.
Yes, that will be part of an enormous dossier, piles of paper, if it were all on paper, that are several feet high, that are submitted to the regulator.
And does it follow that from the provision of information during the trials, that the regulator will have a very good understanding of the levels of diversity in the trials, whether or not there is an untoward degree of bias in the trial process, and whether or not the trials are being tried across all sectors of the population who may ultimately benefit from the product, or perhaps necessarily have to exclude groups such as pregnant women or the immunosuppressed or those with comorbidities.
The MHRA will be aware of all that, yes.
And in relation to the COVID-19 vaccines, in terms of diversity, do you have a view as to the degree of diversity?
In nearly all trials, the degree of diversity is not ideal for a whole host of reasons. But in terms of efficacy, it is very rarely the case, extremely rarely the case, that major groups will have different efficacy, though that tends to be more of a case with young children. Young children are not young adults, they will not behave in the same way. And so their response may be quite different, and you may need to have trials in them. But if I have trials in, let us say, people from Southampton, then they're likely to be similar results to London, and there'll be similar results to Paris, and there'll even be similar results to Cape Town in terms of efficacy. With safety, it is a little more difficult. In general, they will be similar, but not quite as they're certainly not identical. So diversity in the trial for efficacy reasons is not usually a major consideration. It becomes more of a consideration when harms might be associated with different groups.
In relation to the ethnic minority communities, in general terms, did any of the 3 COVID-19 UK vaccines give rise to different results, different outcomes during the trials in terms of the risk of adverse reactions or side effects in the case of ethnic minorities? So,
not as far as I'm aware. Look, one of the problems, of course, is that if you were to be really sure of it, you would need to have 30,000 people from one particular ethnic minority in order to be sure. And that would need to be repeated across others. So we're back to the ethical problem of, do we carry on doing randomised trials with hundreds of thousands of people in them?
And such a trial would be as equally as unrepresentative of the population, which is intended to be benefited.
Yes. So major efforts are made to be diverse, and that's one of the reasons why you may go to multiple centres and multiple countries. So for the AstraZeneca vaccine, a great deal of the data arose from South Africa. In trials in America, the ethnic diversity was they made efforts. It still wasn't representative, but there was considerable effort made to include people from different racial backgrounds.
I want to ask you about that in a moment, but before we get there, and just on the question of the representative nature of the trials generally. So in relation to safety, where there is a real issue in terms of the position of members of the ethnic minorities or ethnic minority communities is not in relation to whether or not the possible impact upon them genetically by virtue of a suspected adverse side effect from a vaccine. It's about confidence. The great need to ensure that trials are representative of the population is to encourage confidence that they are proper trials, that they've adequately got on top of all the safety issues, and that the vaccine is something which can have.
Yes, it's an issue of confidence, but you're also interested in the data. If there's anything startling that you notice, and that's part of what a data monitoring board will do, it will look at subgroups and look at individuals. And so it will raise, hey, we might have a problem here.
But obviously, if you're a pregnant woman or you're a child, there is a greater likelihood that there will be a different outcome in terms of safety than other sectorial groups.
Not necessarily.
Not necessarily. All right. And then the reality of these vaccines, as you say, in respect of at least two of them, there were. Oh, well, in fact, all of them. There were trials outside the United Kingdom.
Yes.
And in relation to, I think, AstraZeneca, there were trials in Brazil and South Africa.
Yes.
And the representative makeup of the trial in Brazil, for example, was 31.4% non-white. And in South Africa, 87% non-white.
So
does it follow that
representative of South Africa?
When debating diversity and whether or not trials are properly representative of the population, one must remember, is this right, that the trials will be conducted outside the United Kingdom as well as inside the United Kingdom? All right. And just finally then, please, on this topic, Professor Prieta Alhambra, at pages 19 and 20 of your report, under the heading of diversity in clinical trials, you say that on this topic of sectorial representation, the initial AstraZeneca trials included a lower than desirable number of elderly people, which led to some criticism about the potential efficacy of the vaccine in older populations. Sex representation was approximately balanced, but pregnant women were, as we've heard, excluded from all phase 3 trials. And most ethnic groups were included in phase 2 trials for all 3 vaccines, albeit in lower proportions than their representation in the overall population. But is that in relation to UK trials?
That is in relation to UK trials, yeah.
But you say, given the rarity of the later identified serious adverse events, it's unlikely that including a higher proportion of people from specific ethnicities would have affected our ability to identify specific safety issues. What do you mean by that latter phrase?
Yeah, I think it's very much in line with what Professor Evans was saying. Given the rarity, the low frequency of these adverse events, we wouldn't still have the numbers to identify them in the trials, even if we had increased the proportion of ethnic minorities in them.
And do you make the point, which I've endeavoured rather inelegantly to put to Professor Evans, that perhaps one of the most important aspects of the need to improve diversity is because it'll improve our understanding of the risk-benefit as well as reduce vaccine hesitancy amongst those underrepresented groups.
That is indeed one of my points in the report.
My ladies,
The
that's
leading
convenient.
question's got the answers.
Yes.
Ultimately.
I'm so sorry. One gets to the stage where I'm afraid one just has to
No, it's all right.
cut to the heart of it.
Not being serious. 25-2, please. Mr. Keith.
Professor Evans, after authorization is granted for a vaccine, in this case the 3 vaccines, presumably there is a massive data which is accumulated and made available based upon or drawn from and reporting on the pre-existing phase 3 trials on members of the public and on the clinical rollout of the vaccine. In the case of the United Kingdom, are there in fact a great deal many data sources reporting on broadly the issue of safety of the vaccines?
Yes.
I'm going to look at a moment on the MHRA's 4 pillars and their post-authorization approach, and I'm not concerned about the manufacturer's obligation to report and to continue to report on safety-related matters. But focusing on the data, are there a number of data management tools across the United Kingdom based very broadly upon GP records, NHS records, the academic and health institutes which carried out the trials, as well as management tools relating to the delivery, the actual immunisation process?
Yes.
In your view, does that data process, however disparate it might appear to be, did it provide a proper, thorough overview or process by which safety-related issues and problems and side effects could emerge?
Some very clever and determined people managed to bring those data sources together to do that, yes.
And did they include observational studies which took place both before and after authorization?
Yes.
How good, how thorough were those observational studies in producing data, accurate data, which would give the regulator and the public a good understanding of the reality of the position?
Non-randomized studies are subject to biases, so in a way that randomized trials are not. The quality in the UK of such studies was as high as anywhere in the world, and it provided enormously important data.
And how important in terms of giving rise to real-world data are electronic health records kept in the United Kingdom?
They're absolutely vital. The general practice system is the bedrock of this, but linking to other records is possible and was done in the pandemic, but isn't always simple.
Is there a national system for the retention of electronic health records, or is there a trans-UK system?
Some of the GP systems cross the different countries. Most are either well represented in Scotland or in England.
Could you tell us, please, something about the Open Safely observational study that was carried out. How important was that in terms of providing useful safety-related information?
I'm biased because I was a participant in that. It went from idea between Ben Goldacre and Liam Smith, who were in Oxford and the London School of Hygiene and Tropical Medicine, to a paper that was published in Lancet subsequently in 42 days. And it was quite extraordinary. But the idea was that based on one of the largest providers of GP records, they decided they could track people who had COVID tests and who had got COVID and be able to look at their death rates. And so this was the first basic paper that was able to do that. They subsequently were able to link to vaccination records to not only death records, but some hospital admission records and a variety of other things. They were never able to link to occupational records, but Open Safely has produced and gone on producing many papers that were of international as well as UK importance. And to get some idea of the scale, properly anonymised and properly produced data, in the case, I think, of tens of millions of people, was accumulated and amalgamated and analysed by this Open Safely process. Is that right? Yes, and the key thing was safely. It enabled the research to be done, protecting any knowledge of any individual participants from any of the investigators. So as an investigator, I couldn't tell whether Mr. Hugo Keith was included in my analysis or not, whether and you may well have been, but I wouldn't be able to identify you at any stage. And the care that was taken to do that is part of the safely. It's not to do with the safety of medicines. It's to do with the safety of the data of the participant. So that requires very clever computing skills to do that.
And is the aim of such a data system to try to identify trends, occurrences, events in terms of the appearance of side effects and safety-related issues which are of significance?
That was one of the objects, yes.
Right. And were there, in fact, a number of other data links? I think you referred to something called the clinical practice data link. There were data links put into place by GP service providers. So there was a mass of data out there.
Yes, Open Safely began with one GP provider and then included the other major ones, such that they had 95%. But Professor Prieto Alhambra was involved in other studies, and in a number of instances, these were done across national boundaries, so that he could talk about a study done in Spain as well as in the UK.
Well, that's extremely helpful because you can now give evidence for him as well. Professor, your report at page 14, if we could have that up, please. At paragraph 3.19, you extol the virtues of the pseudonymized NHS records and linked data, data from the National Immunisation Management System Registry. Did those systems allow quality and timely research on safety to be done?
Indeed, I think that UK NHS pseudonymized records were world-leading in the investigation of safety of COVID vaccines, and the NIMS registry specifically made it possible for us to see a complete exposure to vaccination, which is extremely important to be able to identify people who were or were not exposed to the vaccines.
By comparison to the amount of data accumulated by manufacturers pre-authorization, what can you say about the scale of the data that was accumulated post-authorization?
So post-authorization, we accumulated an order of magnitude of more numbers of patients. The nature of the data and the way it is collected is fundamentally different, though, because authorization data would include randomized controlled trials, whilst the post-authorization data is mostly observational and collected in routine practice, meaning that you need to be more careful with the quality of that data and also with the biases arising from the fact that it's observational and not randomized.
But there was very much more of it.
Yes.
It's obviously a complex system, and there are a number of data sources, were there not, both related to observational studies carried out on the population, but also based upon the records kept of the immunisation process itself. So, for example, you refer to the vaccination management tool in Scotland, and also to NIMS, the National Immunisation Management Service
System,
thank you, in England. In your assessment, were those systems for the accumulation of data efficient and sufficient?
They could always improve. So if you look at my recommendations, I do think that we can make sure that there are systems to link the data more quickly and more efficiently, but they were, as I said, world leading and much better than the data that many other countries had available.
But there are two aspects to which you refer in that context. One is, of course, following the departure from the European Union at the end of the transition period, the United Kingdom no longer sat on, by right, the European Medicines Agency or had direct access to any of its data sources. Do you think that absence of a link, direct link, in the absence of a formal presence at Europe made a difference in terms of the MHRA's oversight of the data which was available, the reality of immunisation, and the safety system?
I believe there was very fluent and, let's say, informal collaboration and conversations that made that transfer of certain pieces of information possible. But of course, not being formally sitting in the same table might have limited access to certain pieces of information. When I then go in my report one by one with the different adverse events, I do not think there was a big impact of that in how things were managed. But clearly, I think a learning for the future should be that we should make sure that we have a more formal collaboration and a better flow of data, if possible.
A second point you make at a different part of your report, page 11, paragraph 3.6, is that there needs to be a system by which the studies known as post-authorization safety studies, PASS, can be organised and managed. And one difference, you say, particular difference between the European Medicines Agency and the FDA, Health Canada, and us, in the form of MHRA, is the availability of a process by which PASS studies can be easily or straightforwardly commissioned. Why does that matter, given the massive data which appears to have been available?
So it matters because generating evidence or knowledge from these masses of data requires very specific expertise, and the regulators, in this case MHRA, need to have that expertise available in-house, but also they need to have access to it through commissioning of these kind of studies. If you look at the examples that I mentioned in my report of, for example, Sentinel for the US FDA, C-Note for Health Canada, or Darwin EU for the EMA, they are structured systems that enable the rapid commissioning and execution of such studies. And I think it would be beneficial to have a similar system here.
All right. That's data. Another important part of this jigsaw is the continuing obligation on manufacturers to produce safety-related information. Professor Evans, is there a general obligation continuing on manufacturers after authorization to continue to make available safety-related information?
Yes, the frequency with which they have to do so changed over time. It's generally, for most medicines, annually, but it became 6 monthly, 3 monthly, and even monthly obligations put on them to update the regulators with their safety information.
And in general terms, do they have to provide not just pre-authorization case safety reports, that's to say particular instances where safety-related issues have arisen, but periodic safety update reports, post-authorization safety study protocols, final study reports, as well as, you said earlier, risk management plans?
Yes.
So a complex and perhaps somewhat Byzantine process of reporting. Does it work?
Byzantine is not the word I would use. I think that it sounds bureaucratic, but in a number of instances it has real clinical and public health benefit. But at times it can be bureaucratic. I'd sympathise with you.
Byzantine was not meant to be pejorative. I deliberately didn't use the word bureaucratic. It's quite complex.
It is.
But overall, in your assessment, does it work in ensuring a high level of confidence that any relevant knowledge in the possession of the manufacturers is brought to the attention of the regulator?
Yes, I think in general it does work.
Then we come on to the obligation on the MHRA to continue to assess safety. And Professor Evans, the MHRA extols the virtues of something called its 4 pillars of review. Without asking you to identify them one after the other, they consist of the yellow card system with an added-on monitoring system, 2 analytical systems by which they assess trends and the theoretical and the hypothetical possibility of things happening, and therefore if something happens they realize it's out of kilter, and also the obligation to continue to assess material provided by the manufacturers. Do, in your assessment, those 4 pillars work in terms of ensuring that everything that can be done to make sure that side effect and safety-related issues are picked up?
In totality, yes.
They say, well, they have 4 elements. One of them is the formal epidemiological studies which are carried out. Are those studies which are carried out, or do they overlap with the sorts of studies that we've been discussing generally in terms of the provision of data?
Yes.
Or are they particular studies directed
Both.
and managed by MHRA?
The MHRA will do relatively few studies of its own. They interact mainly with manufacturers, and they require the manufacturers to do the studies. The manufacturers may then get either independent organisations or academics to do them, and the sort of things that Professor Prieto Alhambra said are examples of those studies. But I think you should understand that there are, to me, 2 important differences, or possibly even 3, in the data. The first is the passive things, the yellow card system, which relies on people in the general public, doctors, health professionals in general, and patients reporting to the MHRA. Then there is a looking at electronic health records in general to look for problems, and then there are specific studies using electronic health records.
Just dealing firstly then with the yellow card system, that passive system, because it requires members of the public to get in touch and say, this has happened to me and I'm reporting it. Did it have an add-on or an additional aspect to it, which was that during the pandemic, the MHRA invited people to put themselves forward for monitoring, having identified themselves in the yellow card system, so that they could be followed up?
Yes.
Do you have any idea of how many people were invited to be monitored and how many people...
Of the order of 1 million.
And how many people in reality agreed to be followed up?
My recollection is it was of the order of 50,000.
So in the context of the issue about the importance of ensuring that the population and the public have confidence in these systems, there is a process by which they can materially contribute to it.
Absolutely.
But in terms of the monitoring system, it wasn't an opportunity that was greatly availed of.
My personal view is that it was not of enormous utility. The yellow card system itself was very useful, but there are biases in that, and the biases in the monitoring system, because it's only 50,000 who respond out of 1 million, are potentially much greater. Electronic health records are a much better source for making decisions on whether an effect that you might have seen in the yellow card system is causal.
Professor Priyata Alhambra, paragraph 3.17, your view is that by and large, the yellow card scheme did allow the MHRA to identify safety issues in a timely manner and to produce swift guidance and documentation. So you think, by and large, it did work. Do you have a view as to the efficacy of the monitoring add-on system?
I think that the yellow card system worked really well, as it has always done, and it's always, or typically always, the main source of the first signals. The monitoring add-on did not add much, in my opinion or in my knowledge.
The second aspect, second pillar, is what is known as the rapid cycle analysis. Could you describe in lay terms what that was? What did the MHRA do in terms of trying to analyse predefined events and draw conclusions?
Rapid cycle analysis was developed probably in America, where they used electronic health records to very rapidly look at whether there was an excess of a particular effect of interest using those electronic health records. And the MHRA adopted that kind of strategy this time. I think that is very useful. It doesn't cover everything, but it covers the things that have risen above the melee that you become aware of.
And the third pillar ecological analysis. So it's said to be proactive analysis of trends within particular populations. What is that? It may, in fact, be part of the third pillar, but it appears to be a separate sort of study.
It's the sort of thing that you may be able to do without individual records. You look at totals of things that are reported through the Office of National Statistics, whether there are death rate trends and things of that kind. It's a very weak pillar. It is a pillar. It's reasonable to look at, provided it's not absorbing lots of resources, but its utility is limited.
And then finally on this topic, you've referred to it already, but could you express your view on the efficacy, the impact and the usefulness of the phase 4 trial process? So to remind ourselves, the trials that took place post-authorization, phase 4 in clinical use, members of the public. How wide were they?
The nomenclature there, I think, is a little bit confusing. Many of the so-called phase 4 trials are not randomized, and I think that the randomized trials gave enormous help in very particular areas, such as whether having a Pfizer vaccine after an AstraZeneca vaccine among adolescents had certain gains or losses. And many of those trials were carried out by academics rather than the industry. I'm not aware of, and they were randomized, and they were supported by academics and the UK system for supporting academic research. I'm not aware of many such trials done by the manufacturers, and I'm not fully aware of, you'd have to ask Dame June Rain as to what impact they had on regulatory decisions. I think for the pandemic, phase 4 trials as such, certainly the randomised ones, had a small effect.
So in truth, the majority of the trials conducted post-authorization concerning members of the public were studies,
Yes, they were not
randomised controlled
and they
trials.
were of enormous value.
Right.
Yeah.
Because presumably they were at scale and therefore were better placed to bring to the attention of the MHRA any safety-related issues. And one last question then, on the topic of signal detection. Professor Evans, may we presume that very sophisticated software and data processes are used by the MHRA and other regulators to try to identify and evaluate trends and the significance of events occurring in the general population?
The analysis of signals, and a signal is noticing that you think it is possible that a particular adverse event is associated with a particular vaccine or drug, and the numbers flag this up. What you need to do is then evaluate that carefully. And I think that the MHRA system of doing that, both in the detection of the problem in the first place, based largely on passive surveillance, with its limitations, I think was done very well.
And is it therefore the conclusion that you reach in your report, overall, the post-authorization safety surveillance, monitoring, flagging up, assessment, evaluative process was a good one?
Yes.
Professor Prieto Alhambra, your opinion?
Yeah, very similar opinion.
And in reality, were they any different in substance or in output and value of use from our European neighbours and the American system?
Very similar, I would say.
Professor Evans, there are then a handful of discrete points which have been raised by the core participants, amongst other people, and I want you, please, just to express succinctly your views on these various points. They've been very helpfully addressed in your report, but it's important that we hear what you have to say about them. Some suggest that the mRNA and also the adenoviral vector technology underpinning, respectively, the Moderna and Pfizer vaccines and the AstraZeneca vaccine are regarded, or should be regarded, or are, and this is not, I think, meant as a compliment, but pejoratively, gene therapies, that in some way they operate to alter the genetic makeup of the body and or are therefore prone to have serious genetic side effects.
Absolutely not.
At paragraph 60, you deal with the issue of the UK reporting of myopericarditis, which you'll recall, of course, was an issue which brought itself to the attention of the MHRA, along with other regulators in Europe.
And not only Europe, it was probably even greater concern in the US and Canada.
And Canada and Israel, was it, or was that?
Yes,
TTS?
I believe that the first signs were seen in Israel.
Do you believe that the MHRA responded in an appropriate manner in terms of substance and time to the emerging reports of myo and pericarditis?
Yes.
You've already spoken about the yellow card system. In an overall sense, do you believe that, subject to such amendments and recommendations as my lady may formulate for its continued use, it is overall an effective system for allowing members of the public to notify the regulator of side effects?
It's probably not as effective for members of the public, but is generally pretty effective for health professionals, and they are the ones who report most of the serious adverse effects. Members of the public will tend to report less serious ones that, in most instances, are of less public health importance. It's the serious ones that are not likely to be seen in the trials. The less serious ones are often seen in the trials, and so we don't, if they're frequent, we know about them.
So the vast majority of reports from members of the public tend to focus on perhaps points of reactogenicity, as opposed to the more serious things, which are, of course, very much fewer in number.
They may well do, and the data seem to suggest that.
The suggestion has been made that it should be mandatory for healthcare professionals to report suspected adverse reactions via the yellow card system. What do you say to that?
Well, I think the answer is in the words you use, suspected adverse reactions. How can it be mandatory to report a suspicion? Are you going to get inside the brain of the health professional to detect that they have got a suspicion? I think you want to make it as easy as possible for them to do so. But I think that the notion of mandatory reporting for health professionals is not good. For manufacturers, yes, they've got to report anything that they receive. But for health professionals who are seeing a patient, I think we want to make it as easy for them as possible. But the evidence we had from the 1990s, where it was mandatory in France for health professionals to report, they reported at a lower rate than the UK, where it was non-mandatory.
Many people have, particularly from the COVID bereaved and injured groups, expressed concern about the operation of the coronial system, which is, of course, wildly out of scope for us. But there is an overlap insofar as, of course, there will be many people who, a significant proportion of those persons who suffered serious side effects, who then died, will have been through, or their families will have been through the coronial process. Do you happen to know whether or not there are any cases in which the coronial system identified and considered deaths, fatalities caused by vaccines?
Yes, they
where
certainly did.
those cases had not already been detected through other means or been within the knowledge of the MHRA?
I don't know whether the cases were. I think, because I don't have knowledge of individual cases, Dame June Rain may be able to answer that. But the effect, the adverse reaction, I am not aware that the coronial system found any new adverse reactions. They reported on things that were already known.
You've already helpfully described the position concerning the impact on Britain's access to the EU database concerning safety. There is a database, is there not, called Vigilance. What impact do you think the lack of access to Vigilance from January 21 has had, if any?
It meant that our immediate knowledge of very rare events was less than it might have been.
From your analysis of the information before the Commission on Human Medicines, the MHRA, the GHSC, the JCVI, and so on, and OCMO, does it appear that any of the European regulators were privy to safety-related information, significant information, when we weren't?
I cannot give you an example.
You specifically addressed the issue of TTS, so thrombosis with thrombocytopenia, so that's thrombosis with thrombocytopenia syndrome. This was an issue which arose in the spring of 2021. Have you reached a view, and we'll look at this in detail with Dame June Rain, but have you reached a view as to the the appropriateness or not of the way in which the MHRA responded to that issue being brought to its attention and the speed with which it responded?
My judgment is that they did. It was one of those things that, as was mentioned previously, could be caused by COVID as well as by the vaccine. And so it becomes quite difficult to disentangle the effects.
Professor Prieto Alhambra, pages 15 and 70 of your report, you also opine on this and will receive, please, your view on that, out of turn with the other conditions which you look at later in your report. What is your view on the appropriateness of the regulator's response and the speed of response on TTS in spring of 2021?
As I explain in my report, I think it was an appropriate response. It was the signal was detected a bit later, but when I say a bit, I'm talking days or weeks. And that could be caused also by the fact that the AstraZeneca vaccine was used in younger populations in continental Europe when compared to what we were doing in the UK, and that led to the earlier identification of that signal.
At page 68, Professor Evans, you say something about the public funding of vaccine studies in the context of phases 1 to 3. We had understood that the studies were generally publicly funded, alongside, obviously, the management and the funding by the manufacturers themselves. But is there room for more public funding, in your opinion?
I think the room for more public funding in the post-authorization studies, I think, is key.
So it's phase 4,
Yep.
post-authorization. But the way in which, I mean, it looks as if from these many trials, or studies, sorry, which were carried on, that there was a great deal of public input by way of funding and participation. Are you talking now about post-authorization randomized controlled trials?
I would like to see more of those, so that there
So
were
formal
yes
controlled trials.
And if it's in phase 4, it's likely to be comparisons between, say, Pfizer and Moderna.
Another subject, please. A source of continuing concern, and it's something about which a great deal many views have been expressed, is the question of whether or not vaccines themselves caused excess death. That is to say, whether vaccines killed people in significant numbers. Bearing in mind that there were obviously, in very, very rare cases, a number of fatalities, and in slightly less rare cases, serious side effects. Was there, nevertheless, a general impact from vaccines on overall mortality rates?
As far as one can see, the studies that looked at mortality demonstrated that vaccines reduced mortality and did not increase it in any way. Looking at ecological studies, as we call them, looking at the totals and not being able to identify whether deaths are occurring in vaccinated or unvaccinated individuals is not the best way of deciding whether vaccines are likely to affect the public health. Looking at totals can be affected by so many different things that they are very biased in making causal attribution of effects.
Professor Prieto Alhambra, at your report at page 23, do you look at both the numerous studies which have been carried out, the estimates from a variety of different bodies, but also, as Professor Evans has said, the mathematical models, and conclude that there is best quality evidence, good quality evidence, available to suggest that the vaccines, far from killing people in significant numbers, save millions of lives?
Yeah, all the good quality evidence I could find suggests that indeed the vaccine saved hundreds of thousands or millions of lives globally.
And to get an idea of the scale of your analysis, how many studies, estimates, mathematical models, as well as empirical studies, that is to say, simple reporting of numbers of deaths and rates of deaths compared against rates of vaccination, are there now in the public domain?
So the best evidence I could find includes the cohort studies where one follows a group of people over time and observes how many of them die. So that's at the individual level, and there are many of those suggesting a reduction in mortality, as well as more global analysis of mathematical models that take the estimate of how effective the vaccine is and then compute how many lives were saved by the vaccines. There's many studies in that regard, and the most recent one says that the vaccine saved over 14 million lives globally.
Professor Evans, due to the exigencies of time, I've not asked you in detail about the last final figure by which the safety system is maintained, namely the way in which information about safety is given to members of the public through what's known as patient information leaflets and documents produced by the manufacturers called Summary of Product Characteristics. But in your report at page 73, do you have something to say about the PIL system? And do you observe that one of the problems, one of the rods that we've created for our own back as a country is there are so many ways in which members of the public can get access to information from government websites, DHSE, Public Health England, MHRA, social media, the press, friends, family, whatever, that a way to increase confidence might be to have a single site or a more clearly identified source of specific scientific, medical, safety-related information for individual medicinal products.
That, in a sense, is what the SPC and the PIL do. When there are alternative manufacturers of drugs, they can sometimes give slightly different information because it's under the control of the manufacturer. But for the vaccines, I think that the PIL and SPC are a good way of standardizing that information and ensuring that good information is there.
And then finally, another topic raised by a number of people is whether or not, and this is page 74 of your report, whether or not persons who have reported through the yellow card system could be updated by the regulator or some other government body in respect of emerging safety information related to the particular vaccine upon which they have reported. Do you think that is a sensible way of proceeding?
I think it's impractical. I think it might also discourage reporting because some people wish to report anonymously. And I think trying to have a system that got back to individuals is simply impractical.
And then trying to draw all those threads together, do you make, and there are four I want to focus on, four recommendations at page 76 onwards, Professor Evans. Not, I think, by way of trying to identify overly prescriptively particular things that, in your opinion, need to be done, but perhaps more by way of calling generally for focus to be paid, more focus to be paid to them. A, you extol the importance of having trials that are as sufficiently powered, as large as they can be.
Yes, and I think the trial quality needs to be high. Medicines are not like soap products. People can just put them on the market with very little regulation, but I think we need to have regulations for medicines.
You say at B, there need to be more randomized trials. Is that in the context of the point you made earlier, which was to the effect that post-authorization, there are huge numbers of studies in the population at large, but by comparison, fewer formal randomized controlled trials. They need to be more at that level.
I think we need to make the authorisation of trials easier, and I have been an advocate for introducing vaccines through randomised trials, especially when there's shortage of vaccines, and using what's called cluster trials, so that you randomise Southampton to get Pfizer, let us say, and you randomise Portsmouth to get AstraZeneca, and then study the effects, and you then know that you've randomised and you've got groups that are similar.
And finally, on the question of data, is it your view that whilst there are numerous data sources, and it would seem from your evidence today a profusion of information, I mean, there's a massive data out there, there is nevertheless an unacceptable degree of fragmentation, that there is insufficient linkage between, for example, GP records and occupational records, between the 4 nations of the United Kingdom and their health boards, and between GPs and, for example, hospitals. There needs to be a great deal more thought given to trying to integrate this system to make it work better.
Absolutely. There is a tension between the availability of that kind of thing and personal privacy. And I think we need to have privacy-preserving linkage of data, as is done in Open Safely.
And Professor Prieto-Alhambra, you therefore may have the last word on this point. Do you also say in your report, page 70, paragraph I think I've lost the reference. I think it might have been 6.2, but you say that there needs to be thought given to the more rapid dissemination of data, and again, the drawing together of some of these quite fragmented systems or sources.
Yeah, I think that what I want to recommend is that there is routine linkage of all those data sets, pseudonymized as necessary, of course, but then also with the addition of expertise to analyze that data made available to MHRA on a regular basis.
Thank you very much. Professor Evans, those are all the questions I have for you. Professor Prieto-Alhambra, can we now focus on your report?
I think I might take a break. Is that permitted while leaving Professor Alhambra here?
Certainly. We'll call you if we need you, or come back when you're ready.
We can deal much more shortly with the first part of your report, because many of the areas have been flagged up already very helpfully by you in the course of Professor Evans' evidence. At page 2, you set out your general approach to what you did and what your report is concerned with. The inquiry did not seek to ask you to opine upon the objective safety of any individual vaccine, not least because safety is not something amenable to an objective classification or characterization. Nor did we ask you to opine upon conclusively, in a determinative way, or pharmacoepidemiologically, whether any given vaccine gives rise to any given adverse effect, side effect. Would it have been extremely difficult to express any view on the objective level of risk of a vaccine, or the likelihood of any individual side effect eventuating from its use, given the absence of a conclusive position scientifically, the relatively small amount of time which has elapsed since the pandemic, and also because it is extremely difficult to determine in any one given case whether or not the side effect is actually caused by the vaccine or is coincidental or caused by COVID. Is that a fair summary?
That is fair, yes.
So what you've done instead is, in order to assist those persons who suffered side effects in these very rare cases seriously following vaccination is you've scoured the public domain and the scientific and academic literature which is in it to see what level of evidence there is to support the proposition in every single case, whether or not that particular condition, side effect, appears to be associated with a vaccine. So that they may have some succor, they may know what the reality of the position appears to be, and so the state can see through this inquiry what the scale of the problem, if there is a problem, is. Is that a fair summary?
That is correct. I might add that we tried to focus on those publications and reports that were available during the period of interest to the inquiry, because we thought those would be the ones that were of highest interest.
And if we look at page 8 of your report, are there degrees of quality of the evidence which you found, upon which you then reach a view as to whether or not it may sensibly be said there is actually an association of some sort between the vaccine and the medical condition? So the quality of evidence might be very low, low, moderate, high, or high. And briefly dealing with these first few pages, do you identify the particular type of vaccine, so the mRNA vaccine, Pfizer and Moderna, and the adenoviral vector vaccine, AstraZeneca? If you say yes or no for the sonographer,
yes,
that would help. Thank you. And do you agree with what Professor Evans has said about the lack of genetic consequence of the use of all those vaccines, and that any notion that they have genetic impact is unfounded?
I agree with Professor Evans' statement on that topic.
Paragraph 11, you express your views on the general position about the regulatory structures, and you draw favourable comparisons between us and the EMA, the FDA, and Health Canada, and you've raised the question of the commissioning of past studies. I therefore don't need to deal with that in detail, but at page 13, you make the point that, again, like Professor Evans, safety cannot be characterised in objective terms. It is relative, and it's all about risk-benefit. In terms of the risk-benefit and the obligation of the regulator to determine whether or not the benefit outweighs the risk, by what margin, in your view, must the MHRA be satisfied that the benefit exceeds the risk? Is it an equivocal balance, or must the benefit greatly outweigh the risk?
I think they must be very satisfied that there's a lot of evidence that the benefit outweighs the risk, and I would say for vaccines that's even more so, because of course we are not treating sick people but preventing sickness.
Effectiveness and intended effects. Does the scientific and medical evidence available, which you've reviewed, demonstrate that the COVID-19 UK vaccines were effective at the time that they were authorised?
Yes, and this is one of the few analyses where we have really high quality evidence coming from large phase 3 trials. Is the evidence consistent between all three vaccines, as to their effectiveness and also as to the levels of effectiveness in each case? They are consistent, and they are well above the threshold set out by most of the regulators internationally. What does the evidence say about the levels of effectiveness in specific population subgroups? So is there a massive information of real-world data, that's to say, the reality of the rollout of the vaccines and the intended effects becoming reality? Were the vaccines of less
efficacy significantly in relation to any particular population subgroup?
Not to my knowledge. I think what's important to realize is that some of those subgroups of the population were not included in the trials, but data was generated using these electronic health records and observational studies, and suggested a similar level of effectiveness to that observed in the trials for the other part of the population.
And do you have there particularly in mind pregnant women, breastfeeding women, who were excluded from the trials, but who subsequently, in large part due to data emerging from America, in fact, large-scale data, were offered the vaccines, and do the results and the data thereafter show that the vaccines worked?
Yeah, specifically with pregnant women, they were not underrepresented. They were excluded by design from the trials, and therefore it was extremely important that we had data on the efficacy or effectiveness of the vaccines for them. Also, because they were having very severe outcomes when they got COVID, and indeed the data that I've seen to date, and I know there's more data being published because I've seen it in conferences and the sorts, is all consistent with a high protective effect against severe COVID. Even more recently.
Children were excluded from trials. What does the real-world post-authorization data say about the effectiveness of the vaccines on children?
Children were excluded from the initial trials, but then there were trials conducted specifically for children where we did observe a similar efficacy. So there is trial data on children.
What was the general impact of the variants of the SARS-CoV-2 virus on the effectiveness of the vaccines?
So what the data suggests, and I think UK HSA and the equivalents in the developed nations did a great job at this, is that as new variants emerged, the effectiveness against symptomatic COVID declined, also combined with the waning of the vaccines themselves. However, there is very strong data suggesting that the vaccines still work to prevent severe outcomes, poor outcomes, like hospitalization or death.
So they continue to protect against the worst outcomes, death and hospitalization, but were less effective in terms of preventing you from getting infected and being possibly symptomatic.
And potentially also they lost their ability to prevent transmission, we believe, because of this inability to prevent infection in the first place.
Long COVID it may seem self-evident, long COVID was identified once we'd begun to try to deal with COVID and to live with it, as being a medical condition consequent to the virus itself. So it couldn't have been studied in the trials for vaccines. But what does the data and the evidence say now about whether or not the vaccines helped protect against developing long COVID?
Yeah, so indeed, long COVID was not an outcome in the trials, but there is good quality cohort studies and observational studies suggesting that the existing vaccines prevent or help to prevent long COVID through the prevention of infection in the first place, and also through the prevention of the development of persistent or long persisting symptoms. The effects that they might have on people who already have long COVID are less clear.
It is well known that at the end of December 2020, the United Kingdom, against, I think, the trend elsewhere, determined that extending the dosage interval was a good idea, and not least because the prioritisation of the first dose meant more people could get some protection rather than giving absolute full protection to a smaller number of people. Does the data and the literature which you've examined show, is it good quality evidence to the effect that that was the right decision, that the increase in the dosage interval had a generally beneficial impact?
Yes, so I think there's two levels to this. At the person level, most of the trials had a shorter interval between doses than what we did in the UK. But there was at least one trial of the AstraZeneca vaccine where they observed a higher protective effect when the doses were a little bit farther apart. So at the person level, that's all the evidence we had. At the population level, the fact that we could deliver one dose to double the population within a certain time frame has proved very beneficial in terms of limiting the impact of the virus on people's lives. So overall, I think there's good evidence to say that that strategy worked really well.
Transmission of the virus. There are two levels at which one must consider transmission, are there not? Firstly, how likely is it that you're going to get infected? But secondly, how likely it is that you might then infect another person. Is that right?
That's correct.
And did you consider those two issues separately?
They are related, of course, so not completely independent. They are related. But the likelihood that someone might get infected, even asymptomatically, was studied in the trials, whilst the transmission of the virus was not. So the quality of the evidence for the former is higher than for the latter.
But in both cases, what does the evidence and the data upon which the evidence is based generally show in relation to the effect of the vaccines in reducing transmission?
The data suggests that the vaccines reduced transmission, at least during the early days of the vaccine rollout. That might have changed after there were new emerging variants, as I mentioned earlier.
Now, the bulk of your report is on the topic of vaccine side effects. And as you've described, you've looked at the literature and give us an idea of the nature of the exercise that you undertook. You and your colleagues, and you were greatly assisted by your colleagues in this exercise, how much material have you looked at here, Professor?
We read hundreds of published manuscripts and tens of reports from different regulatory authorities.
And presumably covering the whole scope of vaccine side effects, of which, of course, there are very, very many, to be measured in the hundreds. And I emphasize, not in terms of side effects or medical conditions that have been established to be in any way connected with the vaccines, but side effects which have been brought to the attention of the authorities by members of the population who believe, rightly or wrongly, that they have been associated with the vaccines.
Yes, and unfortunately, our science doesn't allow us to identify a causal association at the individual level, but we tried to focus on those adverse events that we thought could have changed how we managed the rollout of the vaccines.
And so in each case, or in relation to each medical condition, have you looked at the degree of, or the amount of evidence and underlying data which suggests that there is an association with the vaccine? You've identified how much material suggests that the condition may in fact also, or alternatively, have been caused by the COVID virus itself. And you've also opined on, or you've expressed a view on, whether in light of all that material, as it was known about at the time, the MHRA responded in an appropriate way when being informed of the problem.
That is correct. I think when it comes to the quality of evidence, we tried to combine the quality of the studies that we identified with their consistency. Because if you had good quality studies, but they were inconsistent or had conflicting results, then of course that would tell you there is less evidence for an association.
So if we could have, please, page 32 of your report up on the screen. My lady, unless you were wishing to deal with it another way, I'm going to focus exclusively on the very helpful summaries for each condition that Professor Prieto Alhambra has identified. The summaries, you've drawn for our benefit the text and the substance of your report, which runs to pages 30 on to 80 or thereabouts, into summary boxes for each of the conditions, so for ease of understanding. Myocarditis and pericarditis. you conclude that there was moderate high quality evidence suggesting association between those conditions and the Pfizer or Moderna vaccines. And it's or because they're both mRNA vaccines. Is that
Correct. That is correct.
In terms of the severity of outcome, this, what you describe as as a rare condition, a rare adverse event, gave rise to some cases of hospital admission. Is there any material to suggest that there was anything other than extremely rare fatality, or indeed some studies suggest no associated fatality?
There's many reports of myocarditis or pericarditis following the vaccines with very few fatalities. And of course, it's hard to know whether those were related to the condition in the first place or not.
But the studies themselves don't talk in terms of even there being the possibility of large numbers of fatalities being associated with the mRNA vaccines. The evidence and the data, even at the highest point, puts the number of possible fatalities at a very low level.
Very low number indeed.
Right. And does the data suggest, in fact, that paragraph 5.16, that whilst there is a risk of myocarditis after vaccination, the risk was much lower compared to that seen following COVID infection in the unvaccinated? And I should have said that's the conclusion from one particular study to which you refer. Was that outcome, that proposition, consistent with the generality of the material you looked at?
That is correct, and I would say this study in particular by Patone et al. Is probably one of the best quality studies on this topic.
And turning then to the system response and the way in which the condition of myocarditis and pericarditis emerged, do you reach the view that the MHRA's response was appropriate given the nature of the safety signals which were then emerging?
Indeed, I think it was appropriate, partly because both the virus you were preventing with the vaccine and the vaccine itself could potentially cause this condition, but also because, of course, the COVID vaccines also prevented many other problems, including, of course, heart failure and respiratory failure due to the infection. So the benefit risk seemed to be consistent and balanced. And the actions taken were also very much in line with those taken by many other regulators internationally.
Bluntly, if a country is in the course of a vaccination campaign and the vaccine is known and obviously prevents death, in the case of COVID, vaccination prevented deaths on a daily basis and is now recognised to have saved hundreds of thousands of lives in the United Kingdom. If you stop a programme, will people start dying in greater number because they have not got the beneficial effect of the vaccine?
So, of course, the counterfactual of vaccination is, in the case of COVID, with its transmissibility, getting COVID without being vaccinated. And therefore, if you stop a vaccination program like this based on that signal, you would be potentially causing harm.
You're allowing people to die.
Yeah.
So your assessment of the appropriateness of a regulator's response, coming back to Professor Evans' evidence, has to take into account this risk-benefit. You must weigh up the number of deaths and the likelihood of death which would occur if you don't vaccinate against the very rare possibility in the case of myocarditis or hospitalization from the vaccine. That's the balance.
That is correct. There are, of course, other actions that a regulator can take and did take, including making the public aware of this problem so that they could seek care when they felt unwell, or monitoring more closely the data on particular in particular subgroups. For myocarditis, pericarditis, we know that there's evidence that there was a higher risk for younger men after the second dose, for example. So all those things are important, and they do not cause any harm.
TTS, thrombosis with thrombocytopenia syndrome, did the material and the data show that there was an association in rare cases with AstraZeneca?
Yes, again, we have moderate to high quality data suggesting an association.
TTS can sometimes be fatal, can it not?
It is, and it's also said, and there is evidence to say that it was more severe or more fatal at the beginning when we started learning about these effects, and it probably got more treatable as we learned more about it.
How rare is it as a condition?
Yeah, so looking at the yellow card reports from the MHRA, it says it's around 22 per million doses in people below the age of 50, and about half of that in people older than 50.
So 21.8 cases per million and 11.3 per million in those aged 50 or older. And then a proportion of those very, very rare number of cases led to fatalities.
Yes, I think the MHRA estimates are about 1 in 4 overall.
All right. And the quality of evidence, I may have asked you already, showing or demonstrating an apparent association is good or high?
It's very similar to the quality of evidence on myocarditis, pericarditis. So it's moderate high, meaning that we have cohort studies and self-control studies with adequate control for confounding and biases.
Can blood clots, however, be caused by COVID itself?
There is similar quality of evidence suggesting that blood clots, including the rare blood clots of the brain, also known as CVST, can be also caused by COVID disease.
And are your conclusions and is your view similar to the view taken, or rather, is the evidence and the data you've looked at similar to that which is available in Europe, as far as you're aware?
Yes, very similar. This is a point in my report where I think I make a recommendation that a better or more formal collaboration could help, because the fact that the AstraZeneca vaccine was used for a very different population in Europe gave an opportunity for a sharing of data earlier on.
Not every European country adopted a rollout prioritization system in the way that we did, by focusing on the very elderly first, along with their carers, and working down through the ages.
Most of them did, but the AstraZeneca vaccine, as I mentioned earlier, there was less representation of very elderly people in the trials, and some countries like France or Spain, for example, tended to prioritize the use of the AstraZeneca vaccine for younger people because they thought they didn't have enough evidence that it would work in the older populations.
And is that why certain medical conditions started appearing in Europe first? Because generally speaking, AstraZeneca was given to a different age cohort than it was being given in Britain.
I speculate in my report that might be the case, because indeed this condition was more common, as I mentioned, in younger people, and those were the ones being vaccinated with this vaccine earlier on in those countries.
Page 41, Guillain-Barré syndrome. What is that syndrome?
So Guillain-Barré syndrome is an autoimmune and inflammatory condition of the central nervous system that leads to muscle weakness, and it can be life-threatening. It can be quite severe.
What was the quality of evidence suggesting an association with AstraZeneca and, in fact, Janssen?
Again, very similar to the previous, so this would be moderate high, meaning that there are good quality observational studies.
Rare, very rare, or extremely rare?
This one is classified as a very rare side effect.
And can it be caused by COVID itself?
And it can be caused by COVID itself, and there's good evidence on that as well.
Paragraph 5.60, is there a greater risk, indeed, that it may be caused by COVID as opposed to the vaccines?
So again, there's at least a very good quality study in fact two in this case, suggesting that the excess risk is even more following COVID in the unvaccinated people than following the vaccination with the AstraZeneca vaccine.
No trials were stopped or paused on account of the emergence in study cases of Guillain-Barré syndrome. And it wasn't, I think, there was no time, was there, when cases of Guillain-Barré syndrome started to emerge, which led to specific consideration by the MHRA of whether or not to pause authorization by comparison to the debate, which we'll look at with June Rain, about TTS and myopericarditis. Is that right?
I don't think so.
All right.
No, not to my knowledge.
Bell's palsy, page 44, is a rare weakness or lack of movement on one side of the face, so you described it as rare. Rarely serious or fatal, it may not always resolve completely. Is there evidence to suggest an association with the AstraZeneca, Janssen, Moderna and Pfizer vaccines?
Yeah, again, this is what we classified as moderate high quality evidence, meaning that there is good quality observational studies, in this case with a little bit less consistency across them. And
do you have a conclusion as to the comparative levels of risk between getting Bell's palsy from a vaccine as opposed to from coronavirus itself? Paragraph 5.67.
Yeah, there is, again, a number of studies that suggest there is an association between SARS-CoV-2 or COVID and Bell's palsy.
Transverse myelitis. We recall that there were 3 or 4, I think, cases of suspected transverse myelitis in the course of the trials for one of the vaccines. Is it a very rare or extremely rare medical condition?
This sits in somewhere between very rare and extremely rare, with a likelihood or a rate of around or below 1 in 100,000 doses.
No regulator reached the conclusion that the transverse myelitis cases which emerged in the course of the safety trials was in fact causatively connected to a vaccine, did it?
No, there was, as we discussed before, there was the halting of one of the trials in the UK because of a case, but then that was resolved and the trial continued later on.
And is that reflected in the quality of evidence suggesting an association with any vaccine, but particularly AstraZeneca and Janssen? Is the quality of evidence suggesting any association good, bad or low? Or good, very good, good or low?
Here we thought it was low or moderate at best, because there's good quality studies, but they are not consistent. Some of them find an association, some of them don't.
Page 48, immune thrombocytopenia. Is this a condition which in the past has been associated with previous vaccines?
It is, and it was therefore monitored, again, as an adverse event of special interest by most regulators internationally, including by the MHRA.
And post-authorization, has it identified itself as a rare or extremely rare condition?
The data I have seen says it's around 10 per 100,000, so that would be rare.
And is it a condition which can be caused by COVID itself?
It is, again, a condition that we have good evidence can be triggered by COVID itself.
In relation to immune thrombocytopenia, did the expert working group of the Commission on Human Medicines, in fact, give advice on this condition in the spring and autumn of 2021 to the MHRA?
There are discussions in April 2021 on this topic already, yes.
And in light of the risk-benefit analysis and the overall benefits of the vaccination programme, did the MHRA on both occasions determine that vaccination should proceed, albeit notifications and publications concerning the vaccines were amended.
That is correct.
And was that an appropriate course to take in your view?
I think it was an appropriate course, again, in line with that taken by many other regulators, including the neighboring European Medicines Agency. The MHRA, I think, also importantly consulted consultant hematologists and tried to work on an operational case definition to facilitate the monitoring of this condition going forward.
Capillary leak syndrome, you conclude on page 50 that the quality of evidence suggesting association with the vaccines is low. Acute disseminated encephalomyelitis, page 51, an extremely rare condition. You say the quality of evidence suggesting association is low, moderate. And anaphylaxis, which is the severe allergic reaction, page 53, you say the level of evidence suggesting a association is again low quality. So the material doesn't appear in each of those cases to suggest, or at least there's only low quality evidence to suggest that there is an association of any type with a vaccine, let alone causative. Could we have up paragraph 5.119, please? Did you go through all the core participant witness statements and identify a very long list of medical conditions and health issues to which they refer, draw them up in your paragraph 5.119, and then scour the academic and scientific literature for what they said about these conditions and reach a view as to whether or not the literature showed a sufficient degree of association as to merit, in your view, further inquiry?
So we were provided with that evidence, and there's a long list indeed of, I think it's 80 or 81 conditions in my report, for which we conducted a quick review of the literature in PubMed and tried to identify where we could see any evidence of an association, and then go on to make recommendations on which of these conditions could potentially merit further research. I want to add that it is the fact that for some of these there is no published evidence doesn't necessarily mean that there isn't an association. It just means that maybe they are too rare or too complex, or nobody has investigated them. But of course, the absence of evidence that doesn't equate to evidence of absence of an association in this case.
But it says something about the degree or scale of the problem, if there is one. And you identify, if you go back to page 55, 14 of that long list, I think there's 100 and perhaps even 110 conditions that you've looked at, but 14 of them, chronic obstructive pulmonary disease, varicella zoster virus reactivation, i.e. shingles, seizures, shoulder injury related to vaccine administration, tinnitus, autoimmune connective tissue disease, rheumatoid arthritis, systemic lupus. I'm not sure I can go on, really. I'm bound to fall into error in trying to pronounce any more of these. Fibromyalgia, Graves' disease, depression, optic neuritis, and heart failure. You conclude that there is at least some material to suggest an association, and therefore they are conditions which would merit from further investigation by, in particular, the MHRA.
Indeed, I just want to add that some of those, in fact, some of those published manuscripts do suggest no association. So it's not true that all of them had an association. It's just true that there is some evidence or some literature study.
There is at least one study
yes.
that says something about
yes.
the possibility of an association.
Yeah, and in some cases, like for example COPD, that study was neutral, saying there is no association. So I then go on to review each one of them in the summary box in page 55.
But the remainder in that very long list, you found
no
nothing.
Published.
And therefore you don't even suggest that there is a further inquiry into those other conditions.
I do not think, I think we should prioritize the ones where we do see at least some evidence of an association currently.
All right. And then finally, in relation to menstrual disorders, page 63, you say there is no good quality studies to suggest evidence of a long-term impact of the COVID vaccines on menstruation. Page 65, you make what might be to be thought to be a very obvious point, which is that there were multiple mild and temporary side effects identified in the trials, as Professor Evans has said, but also apparent from real-world data, injection site reactions, fever, fatigue, flu-like symptoms.
Correct.
And then there were a number of publicly reported alleged side effects, which, on investigation, you were able to satisfy yourself could not be confirmed. That is to say, they didn't appear to be right. One, that there was any adverse effect on pregnancy. There was no support, no material to suggest any adverse effect on pregnancy.
No good quality data suggesting that, no.
Page 67, no good quality material. In fact, you said no study to date to demonstrate an association between COVID-19 vaccines and fertility in male or female form.
Correct.
Excess deaths, you've already expressed your view on. Thank you very much, page 68. And then you make a number of recommendations at page 70. I just want to pick up just 3 of them. At A, you refer to the regulatory rolling review, which was in place, but you say it should be replicated. B, you make the point you made earlier, as did Professor Evans, there needs to be a better joining up of data to track vaccine risks and benefits in real time. Mm-hmm. D, this is the point you made earlier about PASS studies being commissioned, or rather not being commissioned by us, but being commissioned by the FDA and the EMA. There needs to be a mechanism for the commissioning and funding of specific studies for post-marketing vaccine safety, and perhaps Professor Evans would say, if I can speak for him, of a randomized controlled nature. And then E, you identify the 14 conditions which you looked at and which you suggest there should be further consideration of. And H, there must always be a continuing focus on proper diversity in vaccine clinical trials. Are those the most important of your recommendations?
I think so, yes.
Thank you very much.
Thank you.
Those are all the questions I had.
Thank you. Ms. Morris, I think you have a couple of questions for Professor Evans.
I do, my lady, thank you. Good afternoon, Professor Evans. I ask questions on behalf of the COVID vaccine adverse reaction and bereaved group, just so you understand the context of my questions. And I have 2 questions on 2 discrete topics, please. One about the patient information leaflet and its use at the time of the pandemic. And a second, a more forward-looking question about adverse effect reporting. Okay. So my first topic, patient information leaflets. One of the observations you made in your report was that one study you'd considered found that nearly 20% of individuals had never read the patient information leaflet. And you said that over half of the respondents in that study, 56%, never sought more information about the possible side effects of medicine. So my question's going to be about accessibility and information provided. Just as a point of information, can you assist with, is there any analysis or any studies as to how many of those vaccinated actually received a patient information leaflet? Is that data available?
I'm not aware of that. The UK, as I understand it from one of the manufacturers' evidence, was one of the few states to demand a paper patient leaflet that could be given and made available at vaccination centres. in many instances, there was simply a QR code which enabled somebody with a mobile phone to scan it and go to a website and find the information. From the manufacturer's point of view, that meant that everything was always up to date, whereas a paper leaflet may not be up to date. And keeping a paper leaflet up to date when you're dealing with millions of vaccinations is very difficult. I think it's very likely, and from my own experience, very likely that the majority of patients getting COVID vaccinations did not read a PIL.
Okay. Thank you. But in terms of whether they had them or not, I only asked that because from one of the groups I represent, the evidence that they've provided to the inquiry is that only 19% of that particular group were actually given. And I wonder if there's any data on that, any studies that have been completed?
I'm not aware of that. I think if I may just add a comment, that I would wish to express my enormous sympathy with those who've died following vaccination, and I think there are undoubtedly cases where a vaccine has led to death. There are deaths following vaccination that may not be as a result of the vaccine, but I think as a community, we have to acknowledge that that does happen in extremely rare cases and that such people need to be looked after properly, and their relatives and those who are bereaved need proper treatment. And so anything I've said about those things, I'd want to acknowledge the pain and the loss in those circumstances.
Well, thank you for those comments. And I'm sure they'd extend to those who've been injured by the vaccine.
Yeah, absolutely.
Yeah, thank you. Okay. Just returning back to the information leaflet.
Move on, Ms Morris. So you've asked questions for which I didn't give permission, and if you want to get in the second question, I suggest you ask it now.
Thank you. The forward-looking question I wanted to ask you, Professor, was about the ability for vaccine injury to be reported. One of the things the Vaccine Injury and Bereaved has suggested is that the NHS app, so the app that we can download onto our phones, include a feature that allows individuals to report vaccine injury directly through that app. You've talked a bit about data and connectivity. Is that something which would have your support as a proposal?
There has been an app to report adverse reactions, a yellow card app, since 2015. And so I don't see adding it to the NHS app is a key element. That would lead to complexity. The NHS app is not designed to connect to the yellow card system. Perhaps to have on the NHS app something that tells you about the yellow card app may be more practical. It sounds a great idea, but practicality and existence of an existing one, I would say, is better.
The single point of access is important.
Not necessarily.
Okay, thank you. Thank you, Milady.
Ms Banton.
Thank you, my lady. I appear on behalf of FEMHO, the Federation of Ethnic Minority Healthcare Organisations, and FEMHO is concerned that there is an underrepresentation of Black, Asian and minority ethnic people during pharmacovigilance monitoring, and that raises questions about whether there was underreporting of race-related adverse outcomes in vaccine usage. That's the general premise. Sickle cell anemia is a blood disorder that primarily affects Black, Caribbean and African people. Are you aware of any targeted pharmacovigilance monitoring of the performance of vaccines for this particular subgroup?
I am not aware of a targeted pharmacovigilance activity for people with sickle cell disease or sickle cell anemia. I would say this is a case where I would expect the vaccines to be potentially more beneficial even than for the general population, given the likelihood that these people might get more severe outcomes when they catch COVID. And I think they could and should be included in post-marketing authorization studies.
So you would say that, in your view, there is value in the targeted pharmacovigilance monitoring, like in the sample of sickle cell sufferers?
In the form of post-authorization studies, observational studies, I think I make a generic recommendation in my report. I think it's recommendation I, where I say we should make sure that there is good monitoring of vaccine effectiveness in people who have been underrepresented or literally excluded from trials, like pregnant women, the minute the vaccines are rolled out. And I think this is an illustrative example of precisely that, a group that could benefit from that kind of study being done when the vaccines are approved.
Thank you so much, Professor. If I may, my lady, may I ask the same question of Professor Evans, just on the same point in relation to sickle
I
cell?
don't think we need it, do we? Thank you very much.
Thank you very much, my lady.
I think I've got the point.
Thank you.
Very well. That completes the questions we have for you, professors. I'm really grateful to you for your help. You've obviously done a huge amount of work, and you explained it in ways that I could understand, and I hope that those following could understand. So thank you both very much indeed for all your assistance and for your time today. The internet says 1:10, so I shall return at 2:10.